This is us. We have an age. The age goes up every year. We have a baby. The baby starts from age 0 while their parents continue to age. The child will grow up and the cycle can repeat again. This trick called reproduction is how our species is able to survive through millennia. However, not every species reproduces this way. For some bacteria, which should also have an age, reproduction is more like cloning, in a process called binary fission, where one organism divides into two identical offsprings. But here we have a problem. How should we assign the age of the original organism? The two bacteria are identical, so it would be unjust to assign one as the parent and the other as the offspring. Should we average the age and assign it to both? Well, it turns out a little bit more complicated than that, all in today's Paper Express. Almost 20 years ago, Eric and his friends came across the exact same question. They wanted to know, in organisms without identical distribution of aged and young components between parents and offspring, does aging occur or not? Their model organism of choice is E. coli, which divides symmetrically through binary fission. What they hypothesized is that different parts of a single organism can have different ages. To better understand what happens when the bacteria divide, they took pictures. Here's an example of E. coli dividing. E. coli is a long and thin bacteria in the shape of a rod, which means it has two poles. We can characterize these two poles by editing two different colors. We can see that the pole that has freshly emerged from a division is marked blue, and when a blue pole has lived through one or more divisions, it turns to the color red. Here is a more clear representation of their model. Each pole is not only colored, but also numbered by their quote-unquote age. The newly divided blue pole are always assigned age 0, and when a bacteria undergoes a new round of division, all its existing poles will gain +1 to their ages. How did they test this hypothesis, you ask? Well, they took more pictures. A lot more pictures. They tracked the formation of entire colonies from a single bacteria. 94 colonies and over 35,000 bacteria, with pictures at least every 4 minutes. After all this work, this is what they have learned. Each dot represents a bacteria. After division, the bacteria that inherited the younger pole is marked blue on the left, and the other bacteria that inherited the older pole is marked red on the right. The longer the vertical line, the faster the bacteria is dividing. After several generations, you can clearly see that bacteria that inherited younger poles are dividing at a faster rate. After 9 generations, this effect is very significant and consistent. When we look at how the growth rate is affected by consecutive new pole divisions or consecutive old pole divisions, we can see very clear trends. Where the longer a bacteria carries an old pole, the slower it divides, and vice versa. If we look at the difference in growth rate between siblings who inherit old poles of different ages, we can see the same trend in a different representation. After all, bacteria are not immortal. They just age in a different way than us. And now, we know how. Thanks for watching, and comment down below on what we want to learn next.